Hello and welcome back to Zoology 141. Today we're going to be starting the lecture from Chapter 4 on Tissues, the Fabric of Life. So to start out today's lecture, we're going to talk about what tissues are. That is, we're going to define tissues. We're also going to describe the structural and functional characteristics of each of the 4 different types of tissue. We're also going to briefly discuss the different types of glands that are produced by epithelial tissue and also describe the steps of tissue repair. And finally, we're going to wrap up the lecture by talking about some strategies that you can use as a student to be able to identify tissues in lab. So if you remember back to a couple lectures back when we had the organization of the human body, we said that a tissue is a group of similar cells that are specialized for a particular function or purpose and usually have a similar embryological origin. Now, we're not going to spend much time talking about embryological origin in this lecture, but you should consult your textbook. Basically, there are 3 different types of embryonic tissue, endoderm, ectoderm, and mesoderm, and each of these can become different types of mature tissues within the body. For example, ectoderm can go on to become both epithelial tissue and also nervous tissue, whereas mesoderm is responsible for creating connective tissues and also muscle tissue. Okay, we're going to start out the lecture with some vocabulary that you'll need to know in order to understand the subsequent slides. Histology, first of all, is the study of tissues, whereas histopathology is the study of disease processes in tissues. As you probably know, a lot of times when you have a patient that's come to the hospital or come to the doctor's office, let's say with an unusual mole or something like that, that may appear to be cancerous, they might do something called a biopsy. A biopsy is just a way to remove a small piece of the tissue without killing the organism and being able to look at that through histological analysis. Now we're going to talk about at the end of the lecture the specific types of processes that we do in order to take something from a biopsy and finally take it all the way through till it's on a microscope slide. And this process is called microtechnique. And of course, microscopy is the process we use to actually visualize the cells, tissues, or whatever else we're looking at on that specimen. So tissues can be classified into one of 4 major categories, and these include epithelial tissue, connective tissue, muscle tissue, and nervous tissue. And we're going to talk about each of these tissue types in some detail. Okay, now that we've gone through all 4 of the major tissue types, we're going to take a closer look at epithelial tissue. As I said before, the prefix in the name epithelial means on top, and so epithelial tissues can be found on body surfaces, for example in the skin. They're also found lining the internal body cavities, for example the GI tract, and the functions of epithelial tissues can be very different depending on where they're found. Epithelial tissues that are found on the outside of the body tend to have a very protective function. They tend to be very thick, consist of multiple layers, and the purpose is to prevent the underlying layers from being damaged or abraded and also from drying out. Another function of epithelium is absorption, secretion, and excretion. And for the most part, these 3 functions tend to be more prevalent on the inside of the body. For example, the small intestine has cells in its epithelium that are geared towards absorption of nutrients from the food that you eat, and they also secrete acids and enzymes to help speed up that process of digestion. And finally, excretion is also a very valuable function of epithelial tissue. And this happens primarily within the kidneys, where we basically get rid of excess ions and waste products and take those from the bloodstream and put them into the filtrate that eventually ends up in the urine. Now, epithelium is a very unique type of tissue. Compared to the other 3, it is the most cellular type of tissue. That is, it's composed mostly of cells. The other thing that makes it unique is that it is avascular. That means it has no blood supply. So all the nutrients that supply epithelial tissue actually come via diffusion from the blood vessels that are underneath the epithelial tissue, and that's usually in the connective tissue. So sometimes the nutrients from the blood and from the body fluids have to diffuse quite a long way to get to these epithelial cells. The other thing you should know about epithelium is that it is very well innervated, and that's basically just a fancy term to mean it has a very good nerve supply. And so if you've ever cut yourself with a paper cut, you know that it doesn't bleed, but it can be very painful. And remember, the reason that it doesn't bleed is because we don't have any blood vessels in the epithelial layer. If that cut did bleed, it meant it's probably a deeper cut that goes deeper down into the connective tissues where we're going to find some blood vessels. The other thing about epithelium is that we can classify the surfaces of cells. For example, each epithelium has an apical surface, and that just means the top surface. On the outside of our body, the apical surface would be the surface that is exposed to the ear. On the other hand, the apical surface inside the body would be the surface that is exposed to a body cavity. And so we sometimes call this the luminal surface. In addition to having an apical or luminal surface, epithelia also have a basal surface. That's basically the bottom part of the epithelium that's sitting on connective tissue. And the connective tissue and epithelial tissue are always going to be separated by something called a basement membrane, which we'll talk about here in just a minute. So here's a schematic of a generalized type of epithelium. And this is actually going to be called a simple columnar epithelium, as we'll find out in a few slides. So we can take a look. We see that it has an apical surface. That's the top part. And this would be the part that would be exposed to the external environment, or in the case of this epithelium, probably to the lumen of a digestive organ. And remember, the lumen was just a fancy word which meant internal opening. Now, in addition to having an apical surface, we also have a basal surface. The basal surface is the bottom layer of the cells, and that is the layer that sits on something called the basement membrane. The basement membrane is an acellular protein layer that is in part secreted by the epithelial tissue as well as the connective tissue that's underneath of it. And so one thing to memorize right now is there's always connective tissue under epithelial tissue. There's a few exceptions, but for the most part, anytime you cut through epithelial tissue, the next thing underneath is going to be connective tissue. And in that connective tissue, we're typically going to have lots of blood vessels. So remember that the nutrients, the oxygen, everything that that epithelium needs is diffusing from the connective tissue up into the epithelial layer. So we classify epithelial tissue in three ways. The first of these ways is by the number of layers. Some epithelia are only one cell layer thick, in which case we say that they're simple. Now, simple epithelium tend to not be very good for protection since they're only one cell layer thick, but they are good for the absorption or passage of gases. On the other hand, a stratified epithelium would be an epithelium that's made up of two or more layers. And in particular, the really thick epithelia, like we find in skin, are really good at resisting abrasion and protecting the underlying tissues as well. But they're not as good for absorption or secretion. The other way in which we classify epithelium is by the shape of the cells. And the cells can be classified as squamous, cuboidal, and columnar, and we're going to look at that in just a minute. And finally, we also classify epithelium via the presence of any surface specializations, such as cilia, microvilli, etc. So we're going to look more at these three different types of ways to classify epithelia in the subsequent slides. Okay, we said before that one of the first ways that we classify epithelium is by the shape of the cells. And cell shape of epithelium can be divided into one of three categories. The first of these categories is squamous or squamous, and this just basically means flat or scale-like. And so squamous cells tend to be very, very flattened and very, very thin. And squamous cells can be found on the inside of blood vessels in a single layer, or on the outside of a skin in multiple layers. And remember, we said if it was a single layer, we call that epithelium simple, and if we found it in multiple layers, that would be a stratified epithelium. Cuboidal cells, on the other hand, are cube-shaped, as the name implies, and we tend to find them in areas like the kidney, where we have some secretion and absorption going on. And in general, when there's secretion and absorption, we tend to have at most two cell layers, but usually just one cell layer. So simple epithelia are much better at absorbing and secreting than stratified epithelia are. And so if we're identifying a cuboidal cell, theoretically it should be cube-shaped, but more often than that it looks maybe circular or ovoid. But the big thing you want to watch for here is a nice circular nucleus that's in the middle of the cell. Okay, and finally the third type of cell shape that we see in epithelia is the columnar cells. So these are tall, column-like cells that form the inside of the digestive tract and again often have a secretory function. So take a look at the columnar cells below. You can see that they're in general much taller than the cuboidal cells, but the more diagnostic feature you would notice is that the nucleus here is much more elliptical. It's not round, but it's sort of a jelly bean shape. And so if you're looking in the microscope and you can't decide whether this epithelium is columnar or whether it's cuboidal, take a look at the nucleus, because to me that's the most diagnostic criteria you can use for classifying it as either columnar or cuboidal. And finally, the last way that we classify epithelia is by the presence of any type of surface specializations. And here we're talking about the apical surface, that is the top surface of the cells that are exposed to either air or to the inside of a body lumen. So sometimes we have cells that don't have any specializations, and we just say they're smooth, but other times we have something called microvilli. Now, microvilli are very, very tiny little hills that are in the plasma membrane on the apical surface. And the purpose of these microvilli is to increase the surface area of that cell so that it will be better at secretion or absorption. So we tend to find them in areas where a lot of these processes are going on, for example, within the digestive tract. Another type of surface specialization is something called cilia. And we talked about cilia in the last chapter and basically just said these are little wave-like appendages that help to move mucus and other substances over the surface of an epithelium. The last specialization, which we haven't talked about so far, is something called keratin. Keratin is just a waterproof protein that we find in areas of the body that we don't want to dry out. For example, we have it in the outer layer of the skin on our epithelium, and this keratin layer actually consists of 15 or 20 layers of dead cells that have a lot of keratin in there. And again, the purpose of that keratin is to resist abrasion and also prevent the underlying tissues from drying out. So now we're going to look at some examples of each type of epithelium. And for each one of these examples, I'll show sort of the pictorial example on the left and an actual histological example on the right. And what you'll probably realize is they look very, very different. So starting out, our first sample is something called simple squamous epithelium. Now think about what that name means. First of all, simple at the beginning means that it's only one cell layer thick. And so simple epithelium, what were they good for? That's right, they're good for absorption, secretion, but because they're only one cell layer thick, it's easy for diffusion to move things across a simple squamous epithelium. Now, squamous means that we have flat plate-like cells, again, that are very, very thin. And so this also helps us to produce an epithelium that's going to be very permeable to things like gases. And that's exactly the function of a simple squamous epithelium. One of the functions is that we find it in the lungs, where it helps to absorb oxygen, and that oxygen crosses the alveoli, which are simple squamous epithelium, and moves readily into the bloodstream. And because that cell layer is so thin, it's very easy for oxygen and carbon dioxide to move across. Now, what you should remember about simple epithelium, and in particular simple squamous epithelium, is that it's very thin and fragile. Our next type of epithelium is simple columnar epithelium. Again, the name simple means that we only have one cell layer thick, which means it's not going to be very good for protection, but it should be good for secretion or absorption, or both. The columnar here means that we have the tall column-like cells, and how could we tell that they're column-like? That's right, they have that nice elliptical jelly bean nucleus. And so we tend to find simple columnar epithelia in places like the digestive tract, and where we have nutrients that are moving across the GI tract into the bloodstream, which is going to be beneath that epithelial layer within the connective tissue. And sometimes we have ciliated columnar epithelium, and sometimes we have non-ciliated epithelium. So here's an actual picture of a simple columnar epithelium that I got out of the laboratory. So what we can see here is, first you can notice we have a nice layer of simple columnar cells that are going from the lower left-hand corner all the way up to the upper right-hand corner. And that there's a space above that, and then that space is called a lumen. And remember, the lumen is just the open area through which food or other things is going to be flowing. And the apical surface of that epithelium is the part that is exposed to the lumen. In fact, most cases we would call that a luminal surface. And then beneath the cells we see the basal surface. The basal surface is just the bottom of the cells and the part that's sitting on the basement membrane. Now, you can't really see the basement membrane here, but it would be the junction between the simple columnar cells and the underlying connective tissue. Our next epithelial sample is something called simple cuboidal epithelium. Again, simple meaning one cell layer, and the cuboidal meaning that we have nice circular to cube-shaped cells. Again, they don't always look cube-shaped when you look through the microscope. Look at the picture at right. Those cells look more circular to me, or more oblong. But I can tell that they're cuboidal cells and not columnar cells because they have a nice round nucleus that's located approximately in the center of the cell. And so just like other simple epithelia, a simple cuboidal epithelium is going to be really good for absorption and secretion. And one area of the body where we tend to find this is within the kidney. Okay, here's another example of a simple cuboidal epithelium. Again, this one's probably from the kidneys. So what you can see that I've labeled here is the tubule lumen. You can see that there are 3 or 4 tubules that are traveling from the top of the screen to the bottom of the screen, and that the open space in those tubules is called the lumen. And lighting that lumen, we have a single layer of cuboidal epithelia, so we call that a simple cuboidal epithelium, and then that's sitting on a basement membrane. Now, what you can see here is that we have one basement membrane and then another tubule made up of simple cuboidal cells. So a lot of times we may not see hardly any connective tissue between 2 different epithelial layers, particularly if we're looking at an organ like the kidney, which is made up primarily of tubules composed of epithelial tissue. So now that we've gone through some examples of simple epithelia, let's take a look at some stratified epithelia. Now remember that stratified meant that we had more than one cell layer thick. So this is an example here of a stratified squamous epithelium. And stratified means, again, several layers, and squamous means that we have those flat plate-like cells on the top layer. Now this is something that you need to remember, is that oftentimes in a stratified epithelium, the bottom cells or basal cells might be a different shape than the top or apical cells. But we always classify the cell shape of that epithelium based on the cells that are on the top. So look at the picture at right. You can see that the bottom is cuboidal, but the top cells are more squamous or squamous. So we still call this a stratified squamous epithelium because it's the cells on the top that matter for classifying the cell shape. Now, just like any other type of stratified epithelium, a stratified squamous epithelium gives us a lot more protection than a simple epithelium. And areas of the body where we tend to find this, well, we find it in the rectum, we find it in the esophagus, the vagina, and also on the outside of the skin. Any place that's exposed to friction or we have the potential for desiccation or drying out, chances are it's going to be a stratified epithelium because that gives us more protection. Now we said earlier that epithelia could have various surface specializations, and that one of these specializations was the presence of keratin. And keratin is a waterproof protein that also makes that cell layer very, very tough. And so we tend to find a keratinized epithelium in the skin, whereas the upper 15 or 20 cell layers are actually dead cells of compacted keratin. So they make a nice waterproof covering for the skin, and again prevents the underlying tissues from drying out, and it also prevents pathogens from burrowing down into that layer and getting to the underlying tissues very easily. Now, on the other hand, a non-keratinized epithelium would be found, for example, in the vagina or inside the body. This is still an area where we could have a fair amount of friction. I mean, think about sexual intercourse. But it's also an area that's very moist to the touch. And so that moisture is because we don't have any keratin, or we have very little keratin in the surface of that epithelium. And so it keeps the epithelium wet and also helps to reduce friction. Other areas in the body where you have a non-keratinized but stratified epithelium are on the inside of the oral cavity. And so I like to tell students to do this thing is just wash your hand and then rub the outside of your lip with one finger. You can feel it's soft, but it's still very dry. And now move that finger onto the inside of your lower lip. And you'll notice that the environment changes very quickly. It gets very moist inside there. And that's because the type of epithelium has changed. It's still a stratified epithelium, but the epithelium on the inside of your mouth doesn't have near as much keratin as it does on the outside, and that's one of the reasons it's so moist. So in the previous slides we talked about simple epithelium, we talked about stratified epithelium, and now we're going to talk about something called pseudostratified epithelium. So think about what that word means. Pseudo means false, and so a pseudostratified epithelium is going to look like it's made up of several cell layers, but in reality it's not. And what we find with the pseudostratified epithelium is that the cells are merely just different heights, and some overlap each other, but all of them are attached to the basement membrane. And as long as we don't have cells growing on top of cells, we can't say that that cell layer is stratified. And so we call this cell layer a pseudostratified epithelium because it looks stratified, but it's really not. And we tend to find this type of epithelium lining the trachea. Now, another thing you might notice about this epithelium is that it has these surface processes coming out of the apical surface of the cell. And these are actually going to be cilia. And remember, cilia are actually motile little proteins that are moving mucus back and forth. And so their purpose is to basically move the mucus that's being produced by our goblet cells up to the pharynx so it can be either coughed out or swallowed. And so the correct name for this epithelium would be a pseudostratified ciliated columnar epithelium. Wow, that's a mouthful. The last type of epithelium is something called a transitional epithelium. Now, this is a stratified epithelium in that it's made up of several cell layers, but we call it transitional because the cell shape changes based on how stretched it is. And first of all, let me say that one area that we find this is in the urinary bladder. And as you probably already know, the urinary bladder has this amazing ability to stretch and hold lots and lots of fluid. The human bladder can hold upwards of 1 liter or so of fluid. And so when the bladder is really stretched out, the cells are going to look very different than when the bladder doesn't have much fluid in it. So take a look on the right-hand side of the screen, and I want you to imagine a nice bladder that's just been voided. And so it tends to be very, very small, and the cells on the surface of that epithelium are going to look more cuboidal. But what happens when you go to 7-Eleven and you get one of those 764-ounce mega gulps and you drink the whole thing, and your bladder is going to start to expand very rapidly, and as a result, those cells which looked cuboidal initially are going to be stretched and stretched and stretched, and so by the end they're going to look more squamous or squamous. And so we classify it as a transitional epithelium because depending on when you look at it, it may look stratified squamous or it may look stratified cuboidal. So instead, we just call it a transitional epithelium. Now, up to this point, we've been talking mainly about epithelia that cover or line different organs in the body. But there's also epithelium that serve as glands. And glands are basically just groups of epithelial cells that manufacture and discharge some sort of excretion. And so epithelial glands can be classified into one of two different types depending on whether or not they have a duct. And so exocrine glands are glands which do have a duct and secrete things which basically come out on a body surface. For example, take a look at the gland in the bottom left. You can see that we have some epithelial tissue, and that epithelial tissue is connected to the body surface through a duct or tube. And so examples of exocrine glands would be things like sweat glands. When we sweat, the perspiration is produced way down in coiled little tubes deep in our skin, but those coiled little tubes eventually empty into tubes which empty onto the skin surface. Other examples of exocrine glands are things like the sebaceous glands or oil glands in the skin, and also the glands that produce mucus and also acid within the human stomach. On the other hand, an endocrine gland is a gland which does not have a duct uniting it to the body surface. And so on the right-hand side, you can see the endocrine tissue, which is made up of epithelium, and we know that it's secreting something, but there's really no way for that secretion to directly get to a body surface. So instead, the way that endocrine products get to where they're going is basically just by diffusing through the surrounding tissues and intracellular fluid and eventually hopping a ride on the bloodstream. So endocrine glands don't have a duct, but they do produce secretions which hop a ride on the bloodstream and get to various organs in the body. So examples of endocrine glands would be things like the pancreas, the thyroid gland, and even the ovaries and testes. These are organs that produce hormones that are important in regulating various types of body processes, but none of these glands has a duct that will unite it to a body surface, and so we classify them as endocrine glands. Now, in addition to classifying a gland as endocrine or exocrine, we can also classify it based on the way in which it secretes its substrate. So take a look at the lower left-hand side of the screen, and we can see an example of a merocrine gland. A merocrine gland secretes its secretion simply by exocytosis. The cells are manufacturing the substance, let's say if it's stomach acid in this case, or pepsin, which is a digestive enzyme, and those little vesicles of pepsin basically merge with the cell membrane and leave that cell through exocytosis. And so merocrine glands are not damaged in the process of the secretion. They can keep secreting and going on and on and on, and they don't have to repair themselves. On the other hand, if we take a look at an apocrine gland, it's called an apocrine gland or apocrine gland because the apical surface of the cell contains the secretions, and that whole apical surface will actually pinch off and become part of the secretion. Now in this case the cell can survive, but it has to regenerate part of itself because a large part of it was lost in the process of secretion. Now depending on whether we're talking about humans or animals, people will debate whether there are truly apocrine glands in the body. In animals we do believe we have apocrine glands. In humans we're not so sure that the mammary glands are apocrine glands. But any time you have the apical portion of a cell pinched off, that would technically be classified as an apocrine gland. And finally, the last example here is something called a holocrine gland. In a holocrine gland, the whole epithelial cell ruptures and becomes part of the secretion. And so a good example of a holocrine gland would be within the sebaceous glands of the skin. Now everybody knows that sebum is that oily, waxy stuff that causes blackheads and sometimes can be responsible for acne. Well, that material is actually generated from cells that have accumulated lipid and other compounds, and instead of secreting that material, those cells just rupture and themselves become part of that secretion. And so what that means is we have to have a high rate of mitotic cell division within these glands to continue to generate more cells, which eventually can become part of the secretions. So the glands that we looked at in the last slide were exocrine glands, and that they did have a duct, but they were multicellular exocrine glands, that is made up of lots of different cells. We actually do have something that's a unicellular exocrine gland, that is composed of only one cell, and that would be called our goblet cell. And goblet cells secrete mucus, and they tend to be found in great abundance within the digestive tract and also within the upper respiratory tract. And the purpose of this goblet cells is to secrete mucin. Mucin is extruded onto the apical surface of the epithelium. It hydrates with water and makes a nice sticky mucus coating that can be used to help trap pathogens, particulate matter, and so on. And so it's the goblet cells that are responsible for making snot. Okay, so here's a nice photomicrograph of an epithelium that does contain some goblet cells. Now first of all, let's look at this epithelium and figure out what it is. Well, we can see that the cells in there look columnar because they have nice elliptical nuclei, and they appear to be stacked on top of each other, but they're really not. Each of those cells is actually attached to the basement membrane, so in this case it would be called a pseudostratified columnar epithelium. Now the other thing you should notice is that there's a nice furry layer on top of the apical surface, and that furriness is actually the cilia. And remember, the cilia are there to help disperse the mucus that is produced by our goblet cells. And so where are the goblet cells? We'll take a look at those nice clear bubbles that we see, and those bubbles are accumulation of mucin, and so the goblet cell is what's surrounding that. And so goblet cells don't have to be very abundant, but they can secrete a lot of mucus, and once that mucus is extruded onto the surface of that epithelium, again, those cilia are going to move it around and eventually force it, let's say, back up to the trachea or back up to the pharynx, where we can eventually cough that up or swallow it. Okay, our next group of tissues are the connective tissues. Now, connective tissues, I should point out, are never going to be found on a body surface. They're always going to be found buried underneath an epithelial tissue. Now, unlike epithelium, some types of connective tissues, in fact, do have a good blood supply. For example, bone is a connective tissue, very good blood supply. On the other hand, other types of connective tissue, like cartilage or like tendons and ligaments, don't have a good blood supply. And so if they don't have a good blood supply, they tend to heal very, very slowly. The other thing is that some are innervated and some are not. It just depends. Some have a nerve supply, some do not. The big thing that distinguishes connective tissues from any other tissue type is that it contains very few living cells. That is, the majority of the tissue is actually made up of non-cellular or acellular material, and we call this material the extracellular matrix. And we'll talk about that in just a minute. Now, there's two different types of connective tissue. Our connective tissue proper, or what we call fibrous connective tissue, and then there's everything else, which we call specialized connective tissue. So in the last slide we said that connective tissues are made up primarily of non-cellular material. which is true, but there are some cells in there. And we do need to talk about those cells because the cells are very important for helping to secrete and maintain the extracellular matrix. And so the cells that we have are things like fibroblasts. Now, a fibroblast is a type of cell that is secreting the fibers that we find in fibrous connective tissue. In fact, anytime you see the suffix blast on the end of a cell type, you know that that is a young cell that oftentimes is actively secreting something. For example, chondroblasts will secrete cartilage, whereas osteoblasts will secrete bone. Also within connective tissue, we tend to find scattered adipocytes, and adipocytes are just fat cells, and they're there to store energy, give us a little bit of cushioning. And we can also find some white blood cells, things like macrophages. Now, macro just means large, and phage means eating. So macrophages are very large eating cells that are there to gobble up any bacteria or cellular debris that happens to contaminate the area. We also have our B cells or plasma cells, which are there to produce antibodies and ward off pathogens like viruses and bacteria. And finally, we have something called mast cells. And mast cells produce histamines, which are essential in initiating the inflammatory response and also regulating blood vessel diameter. So here's the artist's rendition of what a connective tissue might look like. So we're just taking a look at the cells at this point, and here we see our fibroblasts, remember, that are actually secreting the fibers that are present. We have our plasma cells that are making antibodies and trying to keep us pathogen free. Our fat cells or adipocytes, which are giving us a little bit of insulation and energy storage, and also our macrophages, which are just there to gobble up any sort of cellular debris or bacteria that might get down in there. Now, the other thing that we have somewhere down there, oh, there it is we have cells that produce histamine, and these are our mast cells. And this is important because when a tissue becomes infected or damaged through trauma, one of the first signs of that damage is inflammation. And inflammation causes redness and swelling and tenderness, but it also helps to bring raw materials to the site of the injury so that we can help to start tissue repair. And we'll talk about tissue repair towards the end of this chapter. Okay, so now enough about cells. We already talked about all the cells that are in connective tissue. Now let's go talk about the matrix. No, not that matrix. The matrix I'm talking about here is the extracellular matrix. That is everything within a connective tissue that is not made up of living cells. And in most connective tissues, the matrix is made up of two things. It's made up of fibers, and it's also made of something called ground substance. Now, ground substance can be sometimes more watery or more gel-like depending on the tissue that we're in. More importantly are the fibers. There's three different types of fibers that we're likely to find within the extracellular matrix, or at least within fibrous connective tissues. And these include things like collagen fibers. Collagen is a very strong, tough fibrous protein which gives us a little bit of flexibility, but more it gives us strength. And so we tend to find collagen in the skin, and it makes it very, very good at resisting tearing. The other type of fibers we can find are something called elastic fibers. Elastic fibers are a lot skinnier than collagen fibers, and they tend to stretch without breaking. And so we do find those in skin as well, and they give skin a little bit of resiliency. I like to think about this as the elastic in a pair of tighty-whities. You know, it's just very, very, very stretchy. And finally, we have something called reticular fibers. Reticular fibers interconnect with one another, and we tend to find it in areas of the body where there's a lot of filtration going on. And so we'll talk about examples of each of these three types of fibers in the subsequent slides. Okay, back to the artist's rendition of a connective tissue, but now we're looking at the fibers. So take a look first at the collagen fiber. Here they show it as looking a little orange or beigeish, but in truth they tend to be more white. The big thing about collagen fibers is they're very thick and very strong and a little bit stretchy, but not a lot. And so the collagen is what gives the skin and other areas of the body its strength. Our next fiber type you're going to see is going to be the elastic fibers. And those are the same color as the collagen fibers, but they tend to be more kinky or wavy. And so you can see how these could stretch out and not break, and so they give us some elasticity or suppleness to the skin in other areas where we find lots of elastic fibers. And the third type of fiber, which we may or may not find, is going to be the reticular fiber. Again, reticular fibers are basically going to be little networks or scaffolds on which cells can sit, and we tend to find this in areas of the body where there's a lot of filtration going on, for example, in the lymph nodes. Okay, so now that we've learned about the two different parts of connective tissue, that is the cells and the extracellular matrix, we're going to go on to talk about the different types of connective tissue. Now we have fibrous connective tissue and also specialized connective tissue. We're going to start out first with the fibrous connective tissues, and oftentimes we call these connective tissue proper because they have all the components that connective tissue should. And so fibrous connective tissues can be divided into either loose or dense connective tissues. And we say this based on whether the fibers are densely packed or loosely packed. So here's something in biology that actually makes sense. And so there's three different examples of each of these categories. Our first example of a loose connective tissue is areolar connective tissue. Now, areolar connective tissue is basically made up of loosely woven fibers, and it surrounds organs and is found deep within the subcutaneous layer around skin and muscle. It also surrounds blood vessels. And so if you know anything about the word subcutaneous, you know that means below the skin. And so the subcutaneous layer is a layer beneath the skin that contains some fat cells, but also lots of areolar connective tissue. And it's really, really good at absorbing and storing excess fluids. For example, when you have inflammation going on and you have a lot of swelling, that extra fluid is usually going to be present in the areolar tissue layer. Now, because the areolar tissue layer is very good at absorbing and storing fluids, we can actually give fluids to a patient if needed in the subcutaneous layer. And so if you have a cat or a dog, if you're a veterinarian, you can simply slip that needle down through the skin into the subcutaneous layer but not into the muscle and put several milliliters of fluid in there. Now, we don't tend to do that with humans because usually we can find a vein without much trouble. But if needed, in a pinch, you could actually administer fluid to that patient using a subcutaneous route by putting that fluid in the areolar space beneath the skin. Our next type of loose connective tissue is adipose tissue. And adipose tissue, as you probably know, is fat tissue. The purpose of adipose tissue is, multifold. One, it does help us store a lot of extra calories. So if you eat too much, whether fat or carbohydrate, we're going to store that extra energy as lipid or triglyceride within our adipocytes. And the interesting thing about adipocytes is they tend to be most abundant within the subcutaneous layer of the skin, but we also find them underneath the abdominal muscles in the greater omentum. And the interesting thing about it is we don't think that the adipocytes really divide that much. In fact, we used to think they didn't divide at all. But when you do put on weight, you put on weight by actually increasing the vacuole of fat within that adipocyte. So let's say that you've been on a diet for a long time and you're pretty skinny, but then you let yourself go, you go to the all-you-can-eat pasta bar, and you go out drinking every night, and as you get fatter and fatter, the reason you get fatter is that the adipocytes are actually swelling with lipids, and so they can increase their size a tremendous amount. And this is important because it enables the body to store energy for times of need, but the other function of adipocytes is to insulate areas of the body, prevent us from becoming hypothermic, and also cushion internal organs. We tend to have a very nice capsule of fat around organs that we don't want to move or be jostled around. For example, we have a nice fat layer surrounding the kidneys that helps to hold them in place. Now, most of the fat that we're going to talk about is white fat, and its main reason for being there is to store energy or cushion. We also have a second type of fat called brown fat. Now, brown fat helps to generate heat to maintain body temperature. And we tend to find this more in newborns or very, very small mammals that are more prone to hypothermia. And so your proportion of brown fat goes way down as you get older. Okay, our last type of loose connective tissue is reticular tissue. And reticular tissue is found in organs, for example, the spleen and also within the lymph nodes. And in both these areas, the reticular tissue is basically made up these interconnecting reticular fibers, and the purpose of this mesh network is to help in some way with the process of filtering. For example, one of the things that the spleen does is filter the blood and help to pull out worn out red blood cells. And the purpose of the lymph nodes, of course, is to filter the lymph. And so this mesh network helps with that filtration process, but also sitting in the mesh network, we have lots of white blood cells that are basically sitting there as sentinels watching for any type of pathogenic material that might come through. And so we'll talk more about reticular tissue next semester when we talk about the lymphatic and circulatory systems. Okay, now let's go on to talk about the dense connective tissues. Dense connective tissues have very tightly packed fibers, and they can be divided into one of three categories: dense regular, dense irregular, and elastic. And we're going to look at examples of each of these three categories. So dense regular connective tissue consists of tightly packed parallel collagen fibers, and this tends to be found in areas like tendons and ligaments. Remember, a tendon connects muscle to bone, and a ligament connects bone to bone. And so it's important to point out that these areas are avascular. They don't have a good blood supply, and as a result, they don't tend to repair themselves very well when they're damaged. And so think about a tendon or a ligament. It's very, very strong as long as you pull that or stress it in the direction of the fibers. For example, if you pull the patellar ligament in the direction of the fibers, it's very, very strong. But imagine somebody that's out playing football and somebody hits them from the side. Ow! What does that do to their knee? It can very easily rip and tear those fibers because any force that's coming perpendicular to the direction of the fibers can break them very easily. And so as a result, you have somebody that's incapacitated, and chances are that wound's not going to heal very easily because there's not a good blood supply within dense regular connective tissues. So as a result, most of the time that person's going to need some kind of surgery. Now, our next type of connective tissue is dense irregular connective tissue. Where dense regular had nice parallel layers of fibers, here we have thick fibers, but the fibers are going every which direction. And so this makes dense irregular very good at being stretched from any direction, and it doesn't have that nice parallel fiber type. And so we tend to find dense irregular connective tissue around the joint capsules, where it basically makes up part of the capsule and synovial membrane, and it also is found in the dermis of the skin. So here's a photomicrograph of the skin, and basically looking at what you saw on the last slide, where would you say the dense irregular connective tissue would be? Is it the red arrow, the blue arrow, or the green arrow? Well, if you said the green arrow, you're correct. The green arrow contains a lot of dense irregular fibrous connective tissue. You can see that it has nice thick fibers, but the fibers are oriented every which direction. It seems almost to be like an abstract painting. The other thing you should know about dense irregular connective tissue is that it usually has a very good blood supply, unlike dense regular. And so if we have a cut or damage to dense irregular, it's very easy to repair itself because it has a lot of cells down there that can repair the fibers very quickly because they have a very good blood supply. Now, if you're wondering what the other layers are here, the blue arrow is actually pointing to a little bit of areolar connective tissue, and finally the red arrow is pointing to some stratified squamous epithelial tissue. And we'll take a look at skin more in the next chapter on the integumentary system. Alright, now we're going to shift gears and move on from our fibrous connective tissues to talk about our specialized connective tissues. Now we call these tissues specialized because they don't necessarily meet all the requirements of a normal fibrous connective tissue. Now most of them have an extracellular matrix, but that matrix doesn't always consist of protein fibers, at least visible fibers, and so we tend to call them specialized connective tissues. And these include things like cartilage, bone, and blood. Now cartilage is a connective tissue that is formed through a dense network of collagen fibers and also elastic fibers that are embedded in this chondroitin sulfate ground substance. Now the strength there is due to the fibers, whereas the resilience is due to this watery chondroitin sulfate. In fact, the majority of the volume of cartilage is actually made up of water. And the chondrocytes are the cells that occur within spaces in the matrix called lacunae. And lacunae are just little holes, and a chondrocyte is a cartilage cell. Now, cartilages don't have a blood supply. That is, there's no blood vessels within cartilage, but it may be surrounded by a dense connective tissue membrane called the perichondrium. Remember, peri means around or outside, and chondrium here means cartilage. And so the perichondrium might have some blood vessels, but there's definitely no blood vessels inside the cartilage itself. And there's also no innervation. And what that means for cartilage is that when it's damaged, it can take a long time to repair itself, and oftentimes it just won't repair itself. And so that creates a problem for people that, say, have herniated a disc in their back or maybe torn a cartilage in their knee. Chances are that cartilage is not going to heal because it doesn't have a good blood supply. Now, our first type of cartilage is hyaline cartilage. Hyaline cartilage is the most common and abundant type of cartilage in the body. We find it, for example, in the nose. We also find it in the sternum. And hyaline cartilage is composed of closely packed collagen fibers, but we can't usually see the fibers. What we do see is sort of this glossy, glass-looking material that has holes in it like Swiss cheese. And so the glass-looking material that's sort of gray or bluish is the actual ground substance in the matrix, whereas the holes or lacunae contain the living cartilage cells. And those are usually going to be called chondrocytes. And remember, there's no blood vessels within there, so the chondrocytes are getting their nutrients through diffusion of materials from the perichondrium through the cartilage, and that takes time. The other type of cartilage we have is something called elastic cartilage. Elastic cartilage actually has visible fibers, and a lot of these fibers are elastin, which means they're stretchy. And so elastic cartilage is going to be a lot more flexible than, let's say, hyaline cartilage. So we tend to find it in areas, for example, in the ears that are undergoing a lot of stretching. We also find it in the epiglottis and the larynx. Now, again, think about what happens when cartilage is damaged. If you've ever known anybody that played rugby and played rugby for a long time, chances are they had some damage to their ears, and think about how that heals. Yeah, it doesn't look very nice. If you've ever seen somebody that has that nice cauliflower ear on the side of their head, that is a result of destruction and scarring within the elastic cartilage. It does kind of repair itself, but it doesn't do a very good job of it. So just like any other cartilage in the body, if we break it, if we tear it, we probably need to go to the doctor and think about getting some kind of surgery. Okay, a third type of cartilage is fibrocartilage, and fibrocartilage has a lot more visible fibers. That is, it's a mixture of white fibrous connective tissue and also our chondrocytes. And so it tends to be one of the strongest types of cartilage, but also have some resiliency to it. That is, it tends to give a little bit. And the one place that we find a lot of fibrocartilage is in the intervertebral discs. And this is where it serves as a shock absorber. So you're jumping around, moving from side to side. The purpose of these intervertebral discs is to prevent bone from crushing into bone. And so they act as miniature shock absorbers to prevent one vertebrae from colliding into another. But it's important to realize that they can only take so much. That by the time you hit 30 or 40 or 50, the likelihood that you're going to have one or more of these cartilage discs herniating or rupturing is quite high. And when that happens, that can be very painful because some of that cartilaginous material from the inside that's more liquidy can come out and impinge upon the nerve roots that are leaving the intervertebral foramen. And we'll talk more about this when we get to the nervous system. Our next type of specialized connective tissue is bone. Now, bone has a matrix that is composed of inorganic calcium phosphate. And unlike cartilage, it is very well vascularized, which means it has a good blood supply. Now, as a result of having a good blood supply, it tends to heal very readily when it's broken. Now, within the bones themselves, we'll find canals that contain blood vessels called Haversian canals. Also within the bone, we also have a very good nerve supply. And you obviously know this if you've ever broken a bone because, gosh darn it, it really hurts. Now, unlike cartilage cells, which had to absorb their nutrients through diffusion through surrounding tissues, there are canals called canaliculi that help unite the central blood vessel within an osteon of bone with the outlying cells that are sitting in little lacunae. So just like in cartilage where we had cells that were sitting in little holes called lacunae, we have the same thing going on in bone. The difference is that there's sort of an irrigation canal going from these cells to the blood vessel where the nutrients are located. Now, bone, of course, is a very important tissue in the body. One of the things it does is to protect very delicate organs. Think about your brain. It's encased in a nice cranium of bone. The other thing is that it forms bones that can basically be fulcrums for movement. So think about your radius, your ulna, your humerus. They're all there to help you generate movement. Now, your bone tissue is also a very important space for storing minerals, principally calcium, because calcium is needed for several different things in the body. It's needed for nerve impulses, it's needed for muscle contraction, and if you're like me and probably not getting enough calcium in your diet, your body basically can go to your bones and take a little calcium out of the bone bank. Now obviously we don't want that to happen too often, because if that does happen we end up with less calcium in our bones, which can result in osteoporosis. Now the other thing we should point out is that bone tissue can also be a very important site for blood cell formation. Within the epiphyses of our long bones, we have something called red bone marrow, and red bone marrow is responsible for generating red blood cells, white blood cells, platelets, and so on. And so bone can be classified into one of two different types: spongy bone or compact bone. Now the bone that we looked at in the previous slide was compact bone. It sort of looks like the rings of a tree. It has this osteon with a nice central perversion canal where we have a blood vessel, and this is what we find in the shafts of long bones, whereas spongy bone is found in the ends of long bones. So if you've ever sitting there and eaten a chicken wing and you bite through the end of the long bone, you see that sort of sponge-like. You've just hit spongy bone. And spongy bone has lots of spaces where we have a lot of that red bone marrow, which is responsible for generating blood cells. Okay, pop quiz. What kind of bone is this? Well, if you said compact bone, you're correct. You can tell it's compact bone because we have these nice transverse sections that look like rings of a tree or tree trunks. In the middle of that tree, we can see a nice big opening called the Haversian canal, and that's where our blood vessels and the nerves would be. And the outer lying areas where you see the little dark dots, those are the little lacunae where the little bone cells will live. And the bone cells in this case are called osteocytes. Now we'll talk more about bone tissue in a couple more chapters when we get to the skeletal system. Okay, our last type of specialized connective tissue is blood, and blood is certainly probably the weirdest specialized connective tissue you're going to see. And one of the reasons I think it's so weird is that here the ground substance is primarily going to be liquid, and this liquid is called plasma. Now plasma is made up primarily of water and a little bit of dissolved protein. Unlike most other connective tissues, we don't have any fibers, visible or not, found within the blood unless it begins to clot. So what we do have is within the plasma we have several dissolved proteins that normally just remain soluble within the plasma, but if a blood vessel is broken and we have blood loss, some of those soluble proteins can become insoluble and can therefore help to clot the broken blood vessel and prevent further blood loss. Now also in blood, unlike other connective tissues, we find a lot of cells. And the cells that we find are erythrocytes, leukocytes, and thrombocytes. Erythrocytes, as you probably already know, are the red blood cells, and that's what erythros means, red, and the purpose of these red blood cells is to carry oxygen. The leukocytes are the white blood cells, and their purpose is to basically ward off any type of invaders in the body. And then we also have our thrombocytes. The thrombocytes are there to help assist with blood clotting should we rupture a blood vessel. And we'll talk a lot more about blood and the circulatory system next semester. Our next type of tissue is called muscle tissue, and muscle tissue of course is a contractile tissue, and it's composed of interlacing actin and myosin fibers, and we tend to have three different types of muscle tissue in the body: skeletal muscle, smooth muscle, and also cardiac muscle. Okay, our first type of muscle, and probably the one you're most familiar with, is skeletal muscle. Skeletal muscle is the muscle that we find on our skeleton. When you go to weightlift at the gym, it's your skeletal muscle that you're trying to build up. And so skeletal muscle is made up of very long, large cells that contain hundreds and hundreds of nuclei, and of course lots of mitochondria. Now, contraction of skeletal muscle is usually controlled through conscious efforts. That is, we say it's voluntary. You tell your biceps to contract, your biceps contracts. The other property of skeletal muscle is that it's striated, and we'll talk about what these striations mean in a couple more chapters, but it's a way to help us differentiate it from the next one, which is smooth muscle. So here's a nice picture of a longitudinal section of skeletal muscle. First of all, we can tell it's skeletal muscle because we can see lots and lots of nuclei surrounding very, very long tubes, which are the actual muscle cells themselves. Now, previously we said that cells tend to be very, very small and microscopic, but that's not the case for skeletal muscle. Skeletal muscle fibers or cells can be quite long, for example, several inches long. and that's because they started out as little bitty muscle blast cells that eventually united into one very, very long cell. And so each of these cylindrical things that you see here is an individual muscle cell, and it has hundreds and hundreds of nuclei. The other thing you can notice about it, if you squint and look very closely, is that it has perpendicular lines going through the muscle fibers. And so we say that skeletal muscle is striated. It has striations. And remember that skeletal muscle is under voluntary nervous control. The next muscle type is smooth muscle. As the name implies, we don't have any striations here, so none of the striations that we saw with skeletal muscle. Instead, the cells are very, very small and spindle shaped, and they tend to be organized in a willy-nilly fashion. That is, we don't have parallel bundles of fibers like we saw with skeletal muscle. Now, unlike skeletal muscle, smooth muscle cannot be consciously controlled, and it tends to be found in areas of hollow organs. For example, within the walls of blood vessels, and also within the walls of GI or gastrointestinal organs. And within these organs, it helps with the process of peristalsis, which is just basically the movement of food through the digestive tract. So here's a nice slide of some smooth muscle tissue. What you're going to notice is that the fibers are oriented every which direction. I mean, to me, it looks very similar to dense irregular connective tissue, but I can tell it's not dense irregular because I see lots and lots of those little purple dots. And what are those purple dots? That's right, they're nuclei. And here, smooth muscle cells tend to usually only have one nucleus per piece. So we know that the smooth muscle cells must be a lot smaller than the regular striated skeletal muscle cells. So the big picture is that smooth muscle is found in hollow organs, for example, the intestines, and also in blood vessels, and that it is not under voluntary nervous control. Okay, and our last muscle type is cardiac muscle. If you've taken medical terminology or just watched TV, you know that cardiac means heart, and so cardiac muscle tends to be found only in the heart. Now, cardiac muscle is striated, just like skeletal muscle was, but the cells are very, very short, and they're connected to each other by something called intercalated discs. Now, these intercalated discs allow passage of molecules and electrical signals between cells, which is important because one of the things that cardiac muscle has that skeletal muscle doesn't is it has this autorhythmic potential. That is, some cells within the cardiac muscle can actually generate their own electrical impulses and cause that heart to contract. So here's a good picture of a longitudinal section of cardiac muscle. What you can notice first of all is that each cell only has one, maybe two nuclei, and that some of these cells branch just like a tree branches. The other thing you'll notice is that on occasion you'll see these dark perpendicular lines that are the intercalated discs. These can be really hard to see in lab, and usually we have to treat the tissue with a special stain to see it, but intercalated discs are only found in cardiac muscle, and they allow the passage of electrical information from cell to cell, which is very important in order for the heart to contract rhythmically. The other thing that you might be able to notice if you squint very carefully at the screen is that cardiac muscle, like skeletal muscle, is striated. It has lots of perpendicular lines going through it, and we'll talk about the function of these lines later on when we get to the muscular system. Okay, our last major tissue type is nervous tissue. And as you're probably already aware, nervous tissue is found in the brain, spinal cord, and also the peripheral nerves. There's two different cells in nervous tissue, the neurons and also the neuroglial cells. Now the neurons are the cells that generate action potentials or nerve impulses, and the neuroglial cells are there to support or insulate the neurons. Now, like skeletal muscle cells, neurons can be very, very long, and they consist of a cell body where the nucleus is, and attached to that cell body are dendrites, which will collect information from adjacent neurons, and then leading away from that cell body we have something called an axon. And an axon you can think of as a long electrical cable that connects that neuron either to another neuron, or in some cases, let's say, to a muscle cell. And so we'll talk about how neurons work in a couple more chapters, but do know that the neuron is the one responsible for generating the nerve impulses, and the neuroglial cells are responsible for supporting and insulating them. And so here you can see a picture of something called the myelin sheath, which is produced by Schwann cells. And Schwann cells is a type of neuroglial cell that helps to wrap around the axon and insulate that neuron. Okay, now that we've learned about the four primary tissue types, we can go on to talk about membranes. And because membranes have usually more than one type of tissue involved. The first of these membranes is the mucous membranes, also called the mucosae. Now, mucous membranes line organs that have openings to the body mucous membrane line organs that have openings to the outside of the body. For example, the mouth, the intestines, the rectum, they all have passages to the outside of the body, so they're going to be lined with mucous membranes. And of course, one of the things the mucous membranes does is produce a lot of mucus. So we have epithelial cells in there that are producing lots of mucin, and this epithelial layer could be either stratified or it can also be simple. And beneath that we have a layer called the submucosa, and the submucosa is a connective tissue layer that connects the mucosa to the underlying structures. Now, the next type of membrane is called a serous membrane or serosee. Now, unlike mucous membranes, which lined organs that had openings to the outside of the body, serous membranes line cavities that do not have openings to the outside of the body. So one example of a place where we'd find a serous membrane is lining the inside of the abdominal cavity. And basically, the serous membrane is a continuous sheet that is doubled over to form two layers. And the outside layer is the parietal layer. The parietal layer is the layer that lines the inside of the abdominal cavity. Whereas the visceral layer is a layer that actually covers the organs themselves. Now, some of you, if you ever did any hunting or fishing or things like that, have probably at one time gutted an animal. I know it's gross to think about, but if you have, remember back to what it felt like inside that abdominal cavity. It was nice and slippery and smooth, and the reason it's slippery is because serous membranes generate a little bit of fluid that helps to lubricate the inside of the body cavity and reduce friction between the internal body organs. And one more membrane that I forgot to make a slide for is the cutaneous membrane. The cutaneous membrane is basically the skin, and the skin is composed of a stratified squamous epithelium, which has connective tissue underneath of it. And unlike the other two membranes we've talked about, the cutaneous membrane is dry because it has a very thick keratinized layer. And so the purpose here is not to reduce friction by producing moisture, but the purpose is to reduce drying out by producing a nice watertight membrane on the outside of the body, and that's called the cutaneous membrane. And we'll talk more about the cutaneous membrane in the next chapter on the skin. So now that we've learned about the four different types of tissues and also about the membranes, we're going to spend a little bit of time talking about tissue repair. Because any time that you've cut yourself or mashed your finger with a hammer, you've caused tissue damage. And so we're going to talk about the steps that are necessary to repair that damaged tissue. And the two major steps include inflammation and also repair. So let's imagine first that we've had damage to the skin or the cutaneous membrane. You or your dog has stepped on a nail, and what's happening? You're starting to bleed. And one of the first things that's going to happen in addition to that bleeding is inflammation. Now, inflammation is a non-specific reaction that occurs in response to just about any injury or disease. And one of the first things that happens is vasodilation. The underlying blood vessels that are in the connective tissue swell, which provide more blood to the area. They also become more permeable, which means fluid leaks out into the surrounding areolar tissue. So that's why during inflammation you actually observe things getting bigger. The next thing that's going to happen is clot formation is going to occur. Basically, we have thrombocytes within our blood that will form a blood clot along with some proteins to help limit any further blood loss. And also delivered with all that blood are a lot of our phagocytes. And phagocytes include things like our macrophages and some other white blood cells. And they're there to gobble up any invading cells and also to grab a hold of any cellular remnants that need to be destroyed. And then finally, we're going to have our capillaries return to normal size. That is, they're going to undilate, and the blood flow and fluid leakage will stop. And that will conclude the period of inflammation. So once inflammation has ceased, we're going to start our second part, which is formation of the granulation tissue and epithelialization. So remember that when we got this cut, not only did it destroy epithelial tissue, but it was deep enough to protrude down into our connective tissue. And so we need to repair both of those. And so We call the granulation tissue the tissue that forms, and so the granulation tissue is the tissue that forms beneath the overlying scab or blood clot. And granulation tissue is composed of collagen fibers which are infiltrated with lots and lots of capillaries that have branched off existing capillaries. Because one of the things we want to do is re-establish the blood flow to areas that may have had their blood vessels severed. And so this granulation tissue has lots of blood vessels in order to do that. Now, over time, this granulation tissue is going to be slowly replaced by fibrous tissue as our fibroblasts get in there and reweave basically a fibrous scar between the two damaged areas of tissue. Now, depending on the extent of the wound and the treatment available, it can heal by one of three different processes. The first of these processes is called first intention healing. And basically in first intention healing, the edges of a wound are held closely together in apposition. That could be through a band-aid or steri-strip or even using sutures. Skin normally forms a primary union without the formation of granulation tissue. And so we can have first intention healing even if we're not having suturing or taping or anything like that. And it usually results in little to no significant scarring. On the other hand, if we have second intention healing, the edges of the wound are separated from each other far enough that we can't have epithelialization happening very readily. And so what happens is the granulation tissue, which is there to form the gap, and the connective tissue actually protrudes above the skin surface and forms something that is very ugly to look at, and in animals we call this proud flesh. And the third type of healing is third intention healing. And this is when we have a contaminated wound that we can't suture shut, but we leave it open so that inflammation can occur, and when that subsides and contamination goes down, we can later on go back and reclose that wound in something called delayed primary closure. Okay, so to wrap up this lecture, I'm going to spend a little bit of time talking about some strategies for observing tissues under the microscope in the laboratory class, because it tends to be Before I do that, we just need to refresh our vocabulary. And before I do that, we just need to refresh ourselves by reviewing the vocabulary that I gave you at the beginning of the chapter. That is that histology was the study of tissues, histopathology, looking at diseases in tissues, and the last three are going to be most important here because they describe the steps needed to basically get a tissue sample from the body and get it onto a microscope slide so we can observe it. So biopsy was the process of taking that tissue out of the body. And microtechnique, as we're going to find out, is the series of processes we use to prepare that tissue and get it on the microscope slide. And then finally we do microscopy, where we get out our microscope, we look at those cells and we classify them. We look at those tissues and we classify them. Of course, before we can look at a tissue under the microscope, we actually need to collect that tissue. And in a hospital that's going to be done through the and in a hospital or in a veterinary clinic, that's going to be done through something called biopsy. And biopsy is just a small surgical excision of tissue for histopathological analysis. And so one reason we might do a biopsy is if we have a mole or some kind of growth on the skin that we're worried is cancerous, and so we're going to take a biopsy punch, like you see here at left, and place it against the suspect area, a little bit of downward pressure, and the circular blade there will cut us a nice circular plug of tissue, which we can use for histological analysis. We're then going to pull that tissue out, and we're going to process it for histological microtechnique. Now, microtechnique is basically all the little steps that go on between collecting that tissue plug and when it ends up on the microscope slide. And the first step of the microtechnique process is to take that tissue and fix it in a fixative, and that's usually going to be formaldehyde or something like that. And the purpose of that fixative is to cross-link the proteins in there, keep it from rotting, but also give it some stable structure so that it can go through the subsequent steps. So after we have that tissue fixed for a while, we're going to run it through a series of alcohol and other solvents and eventually embed it in some kind of embedding medium. Most often we use something that's like paraffin. Most often we use something called paraffin, which is just simple wax. And so by embedding this tissue in wax, we've enabled ourselves to cut it at very, very thin. And so by embedding this, now once that specimen's been properly fixed, we're then going to go on and do the embedding. Embedding is just the process of taking that chunk of fixed tissue and getting it into some kind of hard medium that can be sectioned easily. And most often that medium is going to be something like paraffin, which is a type of wax. So we simply run that tissue through lots of different changes of a solvent until we get that specimen into wax, and once it hardens in that wax, we can then section that wax block on something called a microtome. A microtome is just a tool used to cut very, very thin sections out of our chunk of tissue. And once we've done that, we can mount those sections onto a microscope slide and stain that slide so that we're able to visualize the tissues that are on there. Now, in reality, although I've showed it on one slide, in reality, this process is very complicated and can take a day or more to go through fixation, embedding, sectioning, and so on. But I do want you to just get the gist of it so that maybe you'll appreciate how some specimens end up looking different than others by the time we get done with the microtechnique process. Now, once we have our specimen on the microscope slide, we want to decide what type of microscopy we're going to use to visualize it. Now, depending on the type of specimen you're looking at, you may use a different type. Now, of course, once we have a specimen that's been processed, we want to look at that underneath the microscope. And which microscope you're going to use depends on how small the structure is you want to see. And so in anatomy class, we're primarily going to use the light. And so in anatomy class, we're going to almost exclusively use the light microscope. And so in anatomy class, we're going to just use the light microscope. And light microscopy has a maximum magnification of around 1000x. There are some microscopes that can get a little bit more, but we really lose resolution anywhere above 1000x. And so we can use light microscopy to visualize tissues, to visualize cells, and even some of the larger organelles. But generally anything that's less than a half micron in diameter is not going to be very well resolved with light microscopy. For that type of study, we need to look at something using something called transmission electron microscopy, or TEM, and this utilizes an electron beam that can be used to visualize very, very, very tiny structures within a cell or even within an organelle. And so it has a maximum magnification that can be greater than 100,000x, up to 500,000x. So it has a lot more resolving power than a light microscope would. Now, the third type of microscopy is called scanning electron microscopy. We don't use it very much to look at tissues because it's used to look at the surface of a specimen. It doesn't see through the tissues like the other two types do. So this is just a picture to give you a comparison of the two different types of microscopes. On your left, we have our light microscope. I don't know what it's about 16 inches high, weighs about 6 pounds, and can go anywhere from between $500 and $2,000. On the right, we have our transmission electron microscope. Probably weighs a couple tons, probably costs 300, 400 grand, up to $1.5 million. And it's used for resolving very, very small structures within cells. And so again, we're probably mostly going to look at light microscopy when we're talking about doing tissue histology. So one question that I get quite often in lab is, why doesn't my tissue slide? So one question I get from students very often in lab is that when they're looking through the microscope, they want to know why the slide they're looking at, that may be labeled cartilage, doesn't look anything like the slide that... And so one of the most frequent questions I get in lab at this point, and so when it comes to tissues, one of the most frequent questions I get in lab is that a student will be looking under the microscope at something that's called cartilage, and then they're comparing that to something else in their book that's called cartilage, and these things look nothing alike. So they'll always ask, why don't they look alike? And we're going to talk about those reasons in the next few slides. And we're going to talk about the reasons why they don't look alike in the next few slides. So here are some facts to keep in mind when looking through the microscope at slides in the laboratory class. So although you may have a prepared slide that says cartilage on it, it often contains several tissue types. And that's because it's often very difficult for the person doing the prepping to just isolate one tissue from all the other tissues without damaging everything in the process. And so usually what we do is get a chunk of tissue that contains cartilage, we embed it, we section it, and it's on that slide. The other thing that you need to know is that most tissues, once they've been cut in those tiny little slices, are darn near transparent. And for that reason, we're going to add some kind of stain to it to be able to see it under the microscope. And there's all different types of stains, which are all the colors in the rainbow. Mostly we use a combination of acidophilic and basophilic stains. Acidophilic stains are things like eosin, which give a cytoplasm, that is the cellular jelly of the organ, this nice sort of pink to orange coloration. On the other hand, basophilic stains like hematoxylin specifically stain the nucleus, and they give it a very dark blue to purple coloration. The last thing you need to know is that tissue processing may also introduce some anomalies that are really not normally present in the tissue, and these anomalies are called artifacts. And so you need to be able to recognize them and differentiate them from things that are really supposed to be there in the tissue. So one of the reasons that your slide may look a lot different than the one in the textbook is because the slide you're looking at, chances are, has several different types of tissues on it. Now, you'll go into lab, you'll pick up a slide that says hyaline, hyaline cartilage, and you'll think, oh, it must have hyaline cartilage on there. And that should be true, but chances are it's going to have a lot of other tissues on there as well. And the reason is because it's very hard for the person who did the dissection or the biopsy to pull off all the other types of tissues without damaging the sample. And so just because it says cartilage on there doesn't mean you won't have other things on there as well. So anytime you look at a specimen under the microscope, look through there and make sure that you can actually find the specimen you're looking at in there and distinguish it from all the other tissues that might be there as well. So here's a picture of that slide that just said cartilage, and we can see, of course, it has cartilage in there, but it also has something called ciliated columnar epithelium and also seromucous cells. So in reality, we have 3 or 4 different types of tissue all in this one slide that said it was just cartilage. The other thing that can mess you up is that the tissues on your microscope slide may have been sectioned at a different angle or a different sectioning plane than the one that's in your textbook. For example, I've got here are 2 different sections of skeletal muscle. That is, it's the same type of tissue, but the one on the left has been sectioned longitudinally and the one on the right has been sectioned transversely. And so initially they look totally different, right? The ones on the left are really long, they have multiple dots in there, the nuclei. The one on the right, we have sort of hexagon looking things that have dots at each corner. And so it's important that you look at enough specimens to realize what this tissue can look like transversely, longitudinally, and also more obliquely. That in both of these sections we can see that there's a muscle cell and it's surrounded on the outside by these nuclei. So don't let the sectioning plane, so don't get freaked out if so the message here is don't get freaked out if the sectioning plane of your slide is different than what you see in the textbook. Another thing that can be very confusing is that the tissue you learn. Another reason why the tissue under your microscope may not look like the one in the textbook is you may have. Another reason why the specimen you're looking at under the microscope may not look like the one in the textbook is that the two specimens may have been treated with different stains. For example, both of these pictures here show a cross-section or transverse section through a peripheral nerve, which is a type of nervous tissue. The section on the left has been stained with something called osmium tetroxide, whereas the section on the right has been stained with hematoxylin eosin. even though they're the same structure, they initially can look very, very different because the stains that have been used are different. So it's important to realize that the color that you see in a specimen is not diagnostic of that specimen. That is, you can't always see, well, there's a lot of blue in this is nervous tissue, because we can always use a different stain. And so the colors in there are artificial, and they've been added by the stains or dyes that we use, and these dyes can vary within specimens. So it's important that you learn to identify the structure rather than concentrating on the colors. And the last thing that can happen is that the specimen you're looking at under the microscope may have histological artifacts in it. Now, what artifacts are just basically abnormalities that were introduced into the specimen during the processing. For example, if you take a look at the specimen at the top left, you can see that this tissue has a very scalloped appearance to it, which you may think, looks like Venetian blinds, but this is very artificial. The reason this happened is that the knife used on the microtome wasn't sharp enough, and so it kind of created this stutter step as it cut through the tissue. There can also be contamination, as you see at the lower left-hand side. That's a picture, looks like it's through the digestive tract, maybe small intestine, and that long blue thing you see in there is actually just a clothing fiber or cellulose fiber. It got in there inadvertently. It's not part of the specimen. Next thing you could have is something called folding. Basically, the specimen gets cut out as a nice thin sheet, and it should be put on the microscope slide in the same manner. But sometimes when we put that on the microscope slide, it inadvertently gets a little bit folded up, which can introduce these artifact folds into the specimen as well. And finally, there can be something called precipitation going on there as well. Remember that most of these specimens are going to be stained with some kind of dye or histological stain. And sometimes that stain contains impurities, and some of those impurities will settle out in the specimen and make something black that's not supposed to be black, or blue that's not supposed to be blue. So be sure you watch out for precipitate of stain and things like that, because it really can throw you off. So as you probably know, you're going to have to identify over 25 different types of tissues on the upcoming lab practicum. And many students will look at that and say, well, instead of looking through the microscope, I'll just go back and look through the textbook. There's some wonderful pictures of these tissues in here, and I'll just memorize them. But you really can't do that, because the tissues I'm going to use on the exam are going to be from the microscope slides. And very likely they can look different than the tissues that are in the book. And so the best way to learn the tissues is to look at several different specimens of the same type of tissue and look for the variation that can be present there. Because once you can begin to identify the same specimen, even though it's cut a different way or maybe stained a different way, you're on the track to understanding and being able to identify tissues. You'll be on the right track to being able to identify. Okay, and lastly, I just want to say in closing is that the best way to learn. Now, as you probably already know, in the next week in lab, you're going to start to learn. Next week in laboratory class, you're going to start to study tissues underneath a microscope, and that come time for the lab practicum, you'll be responsible for being able to identify, let's say, over 20 or 25 different types of tissues. And that seems like a really big deal. Many students will decide that, hey, I'm not going to use the microscope, I'm going to look in the textbook, because the textbook really has some really wonderful pictures of these tissues, and I'll just make flashcards. And that is a mistake, because on an exam, I'm going to give you actual microscope slides or pictures from microscope slides that we've looked at in class, and chances are they won't look like what's in your textbook. So the only way you'll be able to identify tissues reliably is to look at several examples of each tissue type underneath a microscope and quiz yourself that way. This isn't a place where flashcards are going to help out. The best bet is to spend as much time as possible in lab and outside of lab looking at those slides underneath the microscope and getting an appreciation for the variation in that type of tissue. Be able to recognize it, whether it's transversely sectioned or longitudinally sectioned or so on. You've now reached the end of the lecture on tissues. There will be a few short questions after this lecture just to assess your knowledge of the subject matter.
as normal. These questions will not become part of your grade, but if you score less than 70%, I do suggest you go back and review this lecture again before going on to take this week's Lima quiz.